Good morning, friends. In the last lecture, we have discussed about the genome, chromosome, and genes, and what is the modern concept of gene. In those slides, we have been emphasized especially on the eukaryotic gene structure, in which we have noticed that eukaryotic genes mostly are different from the prokaryotic one in the presence of introns. So these introns are the basically non-coding part we have discussed. Then definitely a question come across our mind, then what's the need of these introns within the gene? If they are non-coding, if they are of no use, then why nature has kept them within the gene? Why nature has interrupted the genes, especially in case of eukaryotes? So, in the early studies, it was reported that introns are useless DNA sequences found within the genic sequences. But after that time, many of the studies have been conducted because nature doesn't select useless. Nature always discard useless thing. It's the prime principle claused within the natural selection hypothesis. So it is very important that billions of the forces do not select a useless phenomena or a useless thing, and that kept continuing since last 4 billion years ago, when the first cell might have been emerged. The clear evidences have been of the 3.8 billion years, but still we can consider that in the billions of years the useless things must have been destroyed till that time. But we know very well they are quite interesting that the introns have been still reported. So we are going to consider basics of the introns, then what are the types of the introns that have been discovered till date, what is the biological significance that has been deciphered till date of the introns, because we don't know each and every aspect of the significance of anything on the earth in the 100% manner or in the full manner. We are continue to strive for such kind of significance so that we can add our significance and we can we can make our concept stronger than the earlier one. And then after we'll discuss the alternative splicing. This phenomena is possible because only presence of introns. If there might have been no intron, there is no alternative splicing kind of situation. So one by one we are going to discuss. In the first case, we are going to consider the basics as we have already reported. Say, if this is a gene, this is the intergenic part, and from here to here is reported as a gene. In this gene, basically there are two parts. One is the regulatory part that has been mentioned here in green, that is also known as promoter. It is the simplest kind of regulatory part that has been mentioned. We know very well GC box, cat box, oct box kind of situation. The upstream elements, so-called upstream elements, have been reported. Enhancer kind of elements have been reported. So all these are covered under the regulatory sequences, but still all the genes are having promoters. That's why we are taking the commonest part of a gene. And from here to here, that means from this exon boundary to this exon boundary, this is called structural gene. That means this is the part against whose RNA would be designed by the RNA polymerase. So this is the part that is to be transcribed into primary RNA. Now, these are the intronic sequences. That means what? The coding part is found interrupted by the presence of introns. Okay, so this is the overall situation that we need to understand. The introns have been first reported in globin genes in case of mammals, and ovalbumin gene in case of chicken. They have been called introns because they are found reported as intervening sequences dispersed within the expressed sequences tagged as exons. So, majority of eukaryotic genes do possess the introns, and the presence of intron is the rare feature in case of prokaryotes. What kind of introns found in prokaryotes, that we will consider in the next slide. So these are the introns. Now we are going to consider, first important point is they are of variable size. They may be of 50 nucleotides, they may be of thousands nucleotides in the length, and sometimes their collective length is greater than that of the total length of exons. So it is pretty interesting. And important thing is also lie in the fact that, for example, there is a gene X. For the gene X, the introns found within that gene are not necessary for its expression. So more important thing is, introns, if are found in a gene X, they are not required for the expression of gene X at all, but they may have different kind of significance that we will discuss later. Anyway, in all the cases, we are going to consider the types of introns. That means what kind of introns are found present and where they are found present. So basically, in mRNA, 3 types of introns have been reported. These 3 types, among these 3 types, the most common one is nuclear introns. These introns are found in the nuclear genes. Okay, and these are the most common one. They are being spliced by the spliceosome. Sometimes these are also called spliceosomal introns. So all these RNA are basically protein coding. Okay, now this is the first discovered intron. Then after scientists found in case of mitochondria, and then after in chloroplast, and also in some of the bacteria, group 2 and the group 1. These group 2 and the group 1 have been reported in mitochondria and chloroplast. Group 1 also reported in mitochondria and chloroplast. In this table, it has been mentioned that group 1 is already reported in rRNA genes. It's correct, but they are also found in group 2, mitochondria and the chloroplast genes. So group 1 and group 2 both are self-spliced. We will discuss in detail that what is the meaning of self-splicing and what is the meaning of spliceosomal splicing. There is a separate chapter, RNA splicing, and we will discuss later on that. So these are the 3 different types of introns found reported in mRNA. A single small intron has been reported in tRNA as well. So that tRNA genes are basically spliced through enzymatic removal. Or this is not appropriate to say splicing here, rather than we should consider enzymatic removal. Splicing means the trans-esterification reaction involvement, and removal means there is no kind of trans-esterification reaction, that we will discuss in very detail in the RNA splicing chapter. So next we are going to consider the biological significance. That is the most important thing. So as the stories were coming across our ears that introns, since of its non-coding genic nature, it is almost useless. This is not the situation. Scientists tried a lot and they found many important things. The first important thing in case of evolutionary significance, it was reported that the introns provide the selective advantage by increasing the rate at which coding sequence in different exons of a gene can resort the recombination. In simple words, what it means? It means because of the presence of introns, recombination rate can be increased. And if recombination is increasing, definitely the new kind of combinations are being selected, and we can say that the pace of evolution is increasing. So for the evolution, introns are very, very important. Secondarily, since the introns are non-genic in non-coding nature, so many of the mutations have been generated there and they have been accepted as such, because there is no harm of those mutations there, because ultimately introns have to be removed. So if any kind of mutation is occurring within the intron, it is of neutral value. That's why the neutral mutation rate is increasing and such kind of mutations are being accumulated. So this is also an important feature, especially to study the evolutionary rate. So introns are very important in the evolutionary studies. So we have completed one important point. What is the evolutionary significance of introns? Now we are going to discuss the different and important story, and that we will discuss after the point number 3. The point number 3 is concerned with the size of the intron. As we have discussed in this slide, that they are highly variable in size, and many of the introns are having thousands of the nucleotide pairs in their length. That means we can say there are many introns whose length are very much. Within the intron, there have been reported some of the genes, for example, for miRNA, for example, for endonucleases, for example, for reverse transcriptases. And all these are reverse transcriptases, and also we can consider transposon. Transposase genes are also found reported in the group 1 and the group 2 introns, because they are also the transposons. So more importantly, transposon and reverse transcriptase beautifully define the phenomena of jumping gene, that means transposition, and they are also contributing in evolution. Similarly, miRNA, reverse transcriptase, endonucleases, they all are having regulatory role for the gene expression. So the thing for which scientists have been debated for a long time, that it is almost useless, the vice versa has happened. This is considered today is one of the most important thing. We didn't know about the significance of introns. That doesn't mean they don't have significance. So this is the most important thing. Another important thing is the introns in many kind of tissues are having the tissue specific promoters. That means within the intron there is the evolution, or there is the origin of a promoter, and a different kind of gene might have been evolved within the tissues. So the diversity in the gene expression is basically provided by such kind of promoters. Similarly, these are the sequences that enhances the accumulation of new mutation. So we have discussed about the basic significance of the introns. Here important thing is alternative splicing. Because of the presence of intron, alternative splicing may happen, and alternative splicing make a gene multi-talented. That means a single gene, depending on the situation, may generate a different kind of product. So what is the alternative splicing? We can consider this phenomena in this particular site. For example, this is a gene consisting of 3 exons interrupted by 2 introns. The normal splicing, when happens, these 2 introns have been removed. For example, transcription occurs, primary transcript contains both of the introns. After that, they have been removed, and exon 1, exon 2 and exon 3. All the information in the coding region have been linked together, and a complete long product protein product has been synthesized. So in one situation, if this gene is generating this one, and in another situation, this gene is generating the smaller product from the same gene, but in the different situation, but in under the different regulation. So such kind of splicing, that means alternative ways of splicing are there. That's why it is called alternative splicing. This product and this product are quite different because this product is designed by the information of exon 1 and exon 3 only. The exon seconds information is not considered utilized here. And approximately 75% of genes do show the alternative splicing. In conclusion, what we can say? We can simply say that alternative splicing is the phenomena that basically generate more than one kind of processed RNA. See, processed RNA. Because the primary mRNA is the same, but the processed mRNA is the different. So, the phenomena of generating more than one kind of processed mRNA from the same kind of gene is called alternative splicing. That means it brings about the diversity in gene expression. So this is the overall biological significance of the introns that we can consider. It increases recombination, it accumulates mutation. That's why it is contributing to the evolution. Similarly, it is contributing to the alternative splicing. And what's the utility of alternative splicing? Alternative splicing have the capacity, or it is a kind of art or the phenomena that can respond immediately to the unwanted or never happened situation. So it may have the potential to answer some environmental catastrophes or the environmental unhappenings. That's why they are contributing to the living organism. That's why such kind of phenomena might have been accumulated among the 75% of genes. Thank you so much. Stay home, stay healthy.